Today we are going to explore a banana. Not just how it looks outside, but also what its cells look like under the microscope. Right now, you can see the whole banana in front of you. Once the peel is removed, you can notice the inside part of the banana. You can see the whitish and light green shades, the fibrous line, and the soft fleshy structure. This white part is the edible part that we normally eat. From here, I am going to take a small sample from the white inner portion of the banana. I am carefully scraping a tiny bit and placing it on a clean glass slide. The small piece contains banana cells that we will observe under the microscope. Now I am adding a drop of water on the top of the sample. The water helps spread the sample evenly and keeps it hydrated, so the cells are visible clearly. I removed the extra water using a tissue paper so that the slide does not flood and the focus stays sharp. I am trying to spread the sample inside this drop of water so as to make a thin part of the sample and not a bulgy one. Now I am taking a clean cover slip and placing it on top of the sample using two forceps so that the air bubbles are not formed inside. And now I am pressing it slightly using my index finger so that the sample becomes thinner and not a thicker one. Now I will be extracting the extra water using the tissue paper. You can also do this by adding only a very small amount of water and not adding excess water than what is required. Now I am repeating the same procedure, but this time it is the inside part of the banana peel. This is the part generally we don't eat, we just throw it away. But let's see what we can see inside this part also. The same procedure I am repeating it. I took a small sample using the forceps and trying to place it on the glass slide now. Adding a small drop of water so that it can be evenly visible under the microscope. And now I am using the forceps to spread the sample so that it can be a thinner portion and not a thicker one. Like I did earlier, now I am taking a clean cover slip and I will be placing it on the sample. Remember to use forceps or use a method so that air bubbles are not formed under the cover slip. And slightly I am pushing it right now with my index finger so that the sample becomes thinner. The next part is to remove the excess water. Here I am using a tissue paper. Now coming to the last sample, that is how the banana peel from outside will look under the microscope. For this, I used my forceps to remove a thin portion of the outer peel. I removed a thin portion from the outer peel, but you can see, even though this looks thin to a normal naked eye, it will be a very thick specimen for observing under a microscope. So I got an idea that I'll be scraping this outer peel using a thin knife so that the sample will become thinner. I tried scraping it and finally I got a very thin sample coming up after scraping. You can see the thin sample as compared to the other part which is thicker. Now after scraping it, I took the small sample by cutting it using this knife and I'll be placing it under the microscope. I carefully placed this cut portion of this outer skin of banana peel using forceps, added a drop of water and I will be placing a cover slip over it now. So this is how the glass slide looks after preparation. We have three samples: the main banana part, the inside part of the banana peel and the outer part of the banana peel. Now let's see how all these things will look under the microscope. Here we are looking at the outer skin of the banana peel under 200x magnification. You can clearly see the surface pattern formed by the peel cells. The cells are arranged in a very organized manner, almost like tiny bricks placed side by side. Notice the texture. It looks rough from the outside, but under the microscope it has a structured pattern appearance. You can also see small variations in colors and shading across the surfaces. Now this is under 450x magnification. At this level, the cell structure becomes much more clearer and more detailed. You can see the boundaries between individual cells more sharply. Some cells appear elongated and some look slightly curved depending on the peel surface. The arrangement is still very systematic, showing how beautifully nature organizes even simple fruit peels. You may notice tiny dots and textures inside the cell. These are internal structures and pigments of the peel tissue. At this magnification, the outer banana peel no longer looks like a smooth surface. It becomes a complete microscopic landscape. Now we are looking at the inner portion of the banana peel under the microscope. This is the layer that lies just beneath the outer yellow skin. Unlike the outer surface, the inner peel cells are softer and are more active biologically. As you look at the screen, you can clearly see the individual banana peel cells. They are arranged closely together, forming a protective layer around the fruit. Inside many of these cells, you can see small round globules. Their number is not very high, but they are clearly visible. These structures are mainly starch granules and plastids, also called amyloplasts. Their job is to store food for the fruit. Bananas are rich in carbohydrates and a lot of this energy is stored inside these tiny granules. As the banana ripens, these stored starch granules slowly convert into sugars, which is why ripe bananas taste sweet. Some plastids also change into chromoplasts, which help in producing the yellow color during ripening. These type of storage granules are not unique to bananas only. They are found in other fruits too. Now we are observing the main white portion of the banana, the part that we usually eat. Inside these cells, you can again notice many round globules, just like we saw earlier in the inner portion of the banana peel. But here, the number of globules is much higher. The cells look almost filled with them. These globules are mainly starch granules stored inside amyloplasts, which are special storage bodies inside plant cells. Earlier, in the inner peel, the number of the granules was very low because the peel's main job is protection. But in this banana pulp, the main job is food storage. So the food stores a large amount of starch here, which is why we see so many granules packed inside the cells. These starch granules are extremely important. This pulp is basically the energy bank of the fruit, meant to feed the developing seeds in wild bananas and to provide nutrition when we eat it. Similar storage granules are seen in other fruits and vegetables also. Potatoes are a classic example with high starch content, and fruits like mango, apple, and pear also show such granules during ripening. Bananas may look simple from the outside, but under the microscope, they are full of organized cells and tiny storage granules. Thanks for watching, and I'll meet you again in another microscopic adventure.